We are waiting for the speaker at 2, so probably he will not be here on time, especially because there's some rain. So we are thinking about just the problem session, instead of being at 3 or 4, I don't remember what was the time, it's going to be at 2. So we organized some data sets so you can just play with, and so you can get, what's that? So there was some, these are kind of big questions that I'm going to discuss now. I mean, some of the questions that people are addressing or trying to answer in the field with the data that we have. So then I'm going to go back. I mean, I'm going to go to these questions. And here we have some papers. For example, the first one, this is something that Arnie was mentioning this morning. And there's this landscape that he was showing different tumor types and different genes that are mutated. So there are some of the figures. So the figure that Arnie was showing with the log of the number of mutations and different tumor types. And then there was this kind of plots that it was like that. This is from the first paper here from a few years ago. And then this is some of the data here. This is the TCGA, is the Cancer Genome Atlas. So this is a project here in the US that they've been sequencing 10,000 tumors. So here is the data from 10,000 tumors. So you can look at the mutations that are there. I suggest, because going to the second problem, to take one of the tumor types, in particular glioblastoma, tumors of the brain. And there are 400 exomes, so they are data from 400 patients. So you can just look at the list. And then I'm going to write there a question that is going to be very specific. So you can just go there and then trying to look at what are the mutations. And if you like P53, you can just look at what are the mutations that happen in P53 there, if there is any potential gene also this of interest, and all these things. So the second question is something that Arnie was mentioning, that is how tumors evolve. So in many of these tumors, we can do longitudinal sampling. So you can sequence the tumor in the very beginning, or sometimes in a condition that is pre-tumor condition, and then later and later and later. And then you have genomic pictures of these tumors. And then the question here is to do some inference on what are the trajectories, if there's a particular pattern. Arnie was mentioning that the mutations, they have an order. So it's non-commutative. So things happen, the mutation has to happen at a particular point. It's nothing that you can just change the order. There's some particular structure there. This is pretty interesting. So here is a list of, I think, around 100 brain tumors where we have before therapy and after therapy. And I'm going to mention these things. And then you can just look at the genes that you identified from the first set, some of the genes that are in glioblastoma. And then you can try to see what happens if there are genes that appear in the very beginning of the evolution, or they appear very late, or disappear. Some of the genes disappear in the evolution of tumors. This is probably one of the hottest topics now in cancer, is this archaeology, so these mutational signatures. So as Arnie was mentioning, there are many causes of mutations. So the mutations is not a uniform process. The mutations happen equally everywhere. So they are chemicals that are given. And these chemicals introduce mutations in specific places in the genome. There, for example, the UV radiation or DNA repair is introducing also some type of damage. Now I can give you the information about a patient where there is 10,000 mutations. And I can ask you, can you tell me what are the causes of these mutations? I mean, now that we have a lot of information from many different patients, can we do archaeology and say, well, this person, I mean, was smoking 3 packs a day for 20 years, and this is the number of mutations that are attributed to tobacco, these ones that are attributed to UV radiation, and these things. So there is now a very active field of trying to do kind of archaeology from genomes. I mean, trying to identify what are the causes. Many of those, I mean, a few of those, I mean, very few of those we understand. How are the mechanisms? Most of them we don't. And it's very interesting from the biology. And also, I think from the technical point of view, more kind of trying to identify what is the best way of thinking about these problems. Here, the data set is 560 whole genome sequences from breast cancer samples. All these things, they are published. So the papers are here on these things. And there are 560 whole genome sequences. Junfei has already pre-processed the data, so you don't have to take all the mutations. It's going to give you just the count of mutations of different types that you have. And then you can try to see what is the structure of these mutations. And then it's already pre-processed. It's a matrix where you have patient, 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 and then types of mutations in each of the columns. And then you can count them. There are 3 mutations there, 25, 200, or something. The last one, it's something that is another very interesting problem that is kind of a new tool that we have now to look at a lot of tumors and things, biological processes that are not tumors. That is single-cell data. This is transcriptome data. So we can just look in, for example, in a tumor, and then you can just look at what are the genes that are expressed in different cells. So and some of the things that are very interesting, the tumors, they are not homogeneous. I mean, some of these things, they happen to be, they are heterogeneous, so different parts of the tumor, they can have different mutations. And also, there are many populations of cells that are around that we know that they are very important for the tumor evolution and how these things developed, that you can just capture these things. And now it's like before, I mean, in all the data in TCGA and all these things, you have only a vector of 2,000 genes, and how many copies do you have, and how many copies of RNA you have there. Here, you have a matrix where you have copies in each of these individual cells. So now it's like having like a telescope. I was describing something is that before it was a point. Now you can see that there's some structure there. And the problem there, this is an open problem, and this is why this problem is, I would say, more interesting and challenging, is that we don't know what is the right way of looking at this structure. So there are simple things that you can start doing. So some dimensionality reduction techniques. And there is a variety of techniques that are in the market. I mean, every week there's at least 1 or 2 papers coming with people from different fields thinking about this particular thing. So there are people coming from physics, many people coming from physics, I mean, thinking about this problem. There are mathematicians looking at this problem as you have a very high dimensional space. You have your data is living in a very low dimensional space, and you want to learn properties about this space with some noise. So can you say something about what is the metric structure, the topology of these spaces? Is there something that you can say about these particular things? There are very interesting kind of mathematical work in this arena. There are people from computer science, I mean, looking, for example, in deep learning. So you want to capture what is the important information there. So you can just look at some autoencoders or that type of a neural network that can tell you something about what is kind of a condensed representation. There are people coming from statistics about imputation and all these things. So we have prepared a couple of data sets. And then one of those is also from gliomas, and the other one is from peripheral blood mononuclear cells. These are both published, so there are papers that are associated with that. And then you can play with the data, and you can try to see what is there. The question here is not going to be find a particular cell that is doing these things. It's going to try to uncover what is the structure and how to think of how recover the structure that is underlying to this data that you have. Okay, so we will be going through that. So I suggest, I mean, not during my talk, but I mean after, if you want to start looking at some of this data, and then people can, I don't know, there are 2 people here, I mean, Junfei and Luis, that have been preparing these data sets, and also they will help also to, I mean, to look at this data. Yeah, these are kind of the TAs in these things, although they did most of the work here. So I'm going to follow up with some of the things that Arnie was mentioning 1 hour ago. And there are some, I've selected 4 problems. I mean, there are many problems. I mean, many problems. Some of those are very interesting. But I selected 4 problems where there are data sets that people can play with. So you can go there and try to see what is there. So let me just recapitulate something like kind of the standard model of cancer evolution. This is this cartoon. I mean, it is not a model. It's more a cartoon of how things work. So time is running here from left to right. And the idea is that we have many cells in our body. They are like 10 to the 14 cells. And mutations happen continuously. I mean, one of the mutations that Arnie was mentioning is the amination of particular Cs in these things. That happens in every cell. And there's a repair mechanism that is dealing with that. But some of these things, they are not correctly repaired, and mutations accumulate everywhere. Different carcinogens, they are going to increase the number of mutations in certain tissues. And at some point, some of these cells here, there's one here that is not projected here, acquires a set of mutations that lead to the first clonal expansion. So this is kind of the oncogenesis, the beginning of the tumor. Tumor is a clonal process, so it's coming from a single cell that expands. And here is this, the descendants of this oncogenic cell. They're in green, in what is that, gray here, and they expand here. At some point, there are other mutations that happen in this cell that leads to faster growth. And then this is going to give rise to this kind of pink thing. And of course, I mean, all these populations, they are going to be kind of distinct. So there are different populations of cells. And we see in many tumors that they are not very, sometimes they are very homogeneous, sometimes they are not. So if you sample from different places of the tumor, you have a related genome, but there are differences that are going to be very important. So then there are some diagnoses. So the person is diagnosed. We are going to see some examples with brain tumors. And then the person is going to the doctor. The tumor can be surgically removed. There's some radiotherapy, some chemotherapy, or some targeted therapy. And many of these, most of these cells, they are taken out by the combination of all these methods. But a few of those survive. So they're usually very strong bottlenecks that happen in these populations. After some point, one of these populations start growing again, become resistant to the therapy, and the tumors come back. I mean, they are relapsed tumors. And usually these clones, they are kind of resistant to some of these things, to some of these therapies. And they can be a further expansion that leads to metastasis, and then the clones, I mean, they get additional mutations that enables them to travel all around the body and do these things. So this is the cartoon of how we understand these things. I mean, this is a very imperfect cartoon. We know that it's incorrect in many different levels, but it's the first approximation to how we can understand how some of these processes. So let me just go again through the cartoon. Again, time is running here from left to right. So mutations happened, and as I was saying, mutations happen in every cell, and usually they don't do anything. So there can be a blue mutation here that happens here, and it's not contributing to the phenotype to growing more in this particular thing. Then there is a red mutation that makes this clone to expand at some point. And then there are other mutations that appear at a certain point, the green and the yellow, and they go like that. Because it's a clonal process, you can just start recapitulating these kind of things. Usually it's called clonal because it's coming from a single clone. Darwinian because usually there is this idea that there is competition between different clones, and the conditions that you have, let's say that you have chemotherapy or just going into a new environment or something, they are selecting for these things. And there is this idea of replacement by fitter clones. So there's a competition of all these clones, and at some point there are some of those that are the winners there. That is highly contingent on different external factors. It can be the treatment. So the treatment can make some of these cells disappear, and other ones don't or proliferate. The immune surveillance, that we will have a day that is looking at how the immune surveillance recognize some of these mutations. So it can be recognizing the blue or the red or some of these things. And things that leads to, for example, going to a new place, so invasion or something like that. So when we are doing genomics, what we are doing is taking pictures of these things. We are taking pictures of kind of a snapshot of these things, and here, and here, and here. And in most of these things, we take all the cells, you dissociate the cells, and then what you're just doing is counting the number of cells that they have a mutation, a red mutation. So for example, here, 100% of these cells, they will have the red mutation, 25 they will have the yellow, 50% the green, and these are the numbers that you get, and we are going to see some of these numbers. So what you are going to see, for example, in this plot, they are going to, in the data that you are going to play with, there are going to be points from here, only one time point, and point from here and there in the problem number 2, where you are going to see how these numbers are going to change. And then you have to deduce what's going on in these cancers. Okay. So there are many questions here, and this is why I think it's so interesting. And it depends on your angle. You can have these things. So, for example, the first questions, they are kind of evolutionary questions. I mean, I was mentioning that the dominant first approximation paradigm is this kind of clonal Darwinian process. So you can start asking questions about how these tumors evolve. So the model that I was describing is this idea that there is the clonal replacement of a dominant clone. So there is a clone, is selected, and then selected a random clone there acquires additional mutations, and then they escape and they grow, and then there's this expansion of different clones within the clone. So this is kind of linear because they basically, the only thing that they do is just getting more mutations to the ones that they have. So it's just moving in a direction. But we know that in...
What does the second part of this sentence mean? I mean, or only...
Yeah, so that's I'm going to mention. So, but we know, and now from many studies, including the ones that we are going to see in glioblastomas, in leukemias, and many other ones, this is incorrect. I mean, this is indeed is misleading, and it's going to be very important because that not every tumor cell is going to be the same, and we suspect that there are some particular cells that are going to be more important for seeding what is the thing that is coming back later. So sequencing the average clone is not going to be representative of something that is coming back later. There are several hypotheses, and I think we have ideas in different tumors that can be different. It can be that, for example, the ones that survive to the therapy, they are in a particular niche. So the average, I mean, it can be, for example, in brain tumors, I mean, they remove the main tumor by surgery, And the ones that they are hidden in some place, they are going to be surviving. Obviously, the ones that you remove, they are not going to do much. But we know also, for example, there are some cells that they are going to be quiescent. So some quiescent cells, they are not very sensitive to chemotherapy. They are going to be quiescent. They are not sensitive to the therapy, and they can replenish. They can just repopulate the whole tumor from a particular set of cells. And then if you sequence the information at some point, you are taking information about the dominant clone, so what is the big population. But it can be that this can be misleading about something that is coming back later. Okay. So in metastasis, the same thing. So what is making a clone that is in the first tumor just moving around and be able to see it in a different place in the tumor? There are certain things that are also very interesting. What are the mutational processes in cancer? We are going to see in the problem sets also, I mean, if we can identify what are some of these causes. And if these are the causes, there's this idea also that clonal heterogeneity is very important. The idea is that you can have tumors that are very heterogeneous, and if you have a therapy that you can kill 99.9% of all the clones, then at least a small population of those can be resistant to the therapy, and they can repopulate the whole thing. So heterogeneity is a very important factor that we have to take into account, and also for practical purposes. So if you have a tumor and you go to the doctor, in many places, they take the tumor, they send it to sequence, and you get a report with the mutation. So you can check it, there's some mutations. But it can be that these mutations, they don't tell you anything about something that is coming back later. And this is something that we are trying to address, depends on the therapy and all these things. Identifying what is the origin, the clonal origin of the tumor that is coming back later is a fundamental question that still we don't understand in many tumors. If you're a doctor and then you're looking at patients, you can get these reports, and you really don't care about these particular questions in detail, although you should, several things. But I think one of the things is how informative. You get a list of mutations. Let's say that you have 10 mutations, 20 mutations. How informative it is. What is the predictive value about how this patient is going to evolve, or even to design treatments. So as Arnie was mentioning, some of these mutations, some of the most popular mutations like P53 or RASes and all these things, there is no treatment for them. But there can be something there that is important, and it's treatment. So what is the predictive value of the information that is the report of the patient, of the doctor to these ones? And this is important because it's going to be very related about how these tumors evolve. If there's already some resistant clones, perhaps this therapy is not going to be efficient, and so on. They can be also stratifying patients, or classifying patients. You take 100 patients here, and then some of those, they can be resistant to the therapy. Some of those, they can be non-resistant to the therapy. And you want to understand what are the genetic factors that contribute to these kind of things. I work with a lot of people in kind of molecular biology, and they are trying to understand what are the specific mechanisms. So now you take 100 patients, and we are going to see these things with some of the gliomas. You are going to sequence them, and then you are going to find what happened during therapy that makes this clone resistant to the therapy, for example. And then in there, you can identify some mutations, and then these mutations in some genes, and these genes, they can inform you about the mechanism of resistance, and they can tell you an idea about potential alternative strategies that you can have for treating this particular patient. So this is kind of a few kind of big questions that with collecting all these kind of genomic data, you can start asking about these things. There are many other ones, okay? So for the purpose here of this hour and a half, we are going to focus on 4 different problems. They are going to be related to some of the things that Arnie was mentioning. The first problem is identifying driver genes, and I'm going to mention what is that, in cross-sectional cancer studies. So as Arnie was mentioning, if you just take a patient and you do a whole genome, for example, typically you can get 10,000 mutations. So we know that most of them, they are not going to be important. Only a small fraction of those, they are going to be probably important. But because they happen to be, like the blue mutation here in the cartoon that I was mentioning here, they happen to be in the cell that gets amplified here because of the real mutation. So you want to find the red one, but the blue one. The red and the blue, they are going to come exactly in the same frequency. So they are going to be traveling with the mutation with the clone that is going to be responsible. So these 10,000 mutations usually that you get in a tumor in a report of a whole genome, or if you are doing exomes, this is most of the data in problem 1 and 2 is going to be exomes, then you get hundreds. So of these 100 mutations, you expect that 5 or 6 that they are going to be important, the rest are not. In the jargon of the field, sometimes mention that these are driver genes. They are the ones that are going to be driving the phenotype. And all the other ones, they are going to be passengers. They are going to be around, and they are going to be traveling with the clone. So now the question, one of the questions here, is now if you just get cross-sectional data. Cross-sectional means that you just take many different patients that they have the same disease. So typical studies is you take 200 patients of those, or 1,000 patients. You sequence all of those. Each of those, they have 10,000 mutations that are going to be different. But some of these mutations could be similar. So for example, as Arne was mentioning, P53 you will find in many of these things. So that's the fact that there are 50% of these patients that they have mutations in the same gene, that's something suggesting that there is something important about this gene. So there are certain ways that you can do these things, but this is still an open problem. I mean, the first thing that people do is counting. I mean, just count how many times do you find the mutation in different patients, and if it's more than a statistical background, then you consider it to be a call, and if not, no. So it depends on how you model the underlying statistics. So the second problem is this one of reconstructing the evolutionary history of tumors in longitudinal studies. So longitudinal is when you just take a different time points, and then most of the data, for example, in the Cancer Genome Atlas is cross-sectional. So you take many of these things. The longitudinal is that when you can get at different time points. Leukemias, there's a lot of work because sometimes they are very easy to sample, so you can get at different time points. In solid cancers, it's a little bit more complicated sometimes. So the problem set here is going to be brain tumors before therapy and after therapy. The third problem is this archaeology of human cancers and this identifying the mutational signatures. So given that you have some mutations, 10,000 mutations in a tumor, can you go and tell, well, this is caused by tobacco, and this is caused by aging, and this is caused by aflatoxin? So what are the different causes that are associated to that? And can you identify, more interesting, can you identify some causes that they are not reported? For example, some chemical compounds that we are not aware of can introduce some mutations and lead into some of these tumors. This is kind of an interesting open problem. And the last one is the single-cell data that I'm going to briefly mention. Again, so here is the web page that you already know that. So I'm going to go to some of the basics of how this sequencing is done, and then what is the type of mutations that, that people look at. And for the problem set, it's going to be only substitutions, that they are going to be the change of one base to another base. It's the simplest one. But I mean, this problem can be done in different things. So this is from a review from Matt Meyerson. So you take a tumor material. So you just isolate the DNA. So you have many cells here. You dissociate, you eliminate the cells. You just get the DNA. And then you just fractionate the DNA. You chop it into pieces. And then you just put it in one of these machines for sequencing. And then you just get information pieces, sentences. They are usually like 100 base long. And this is going to be the information about the tumor. The next thing that you have to do is alignment. So we have several human reference genomes. So you take the favorite one. You just have a base and a scaffold that you are going to use for mapping these things. So typically, I mean, you can have, for example, 1 billion of these sentences. I mean, you can imagine that there is kind of a book, and then you have all these sentences that are in different places, and you have to assemble in a book. You have a template, so you can be aligning these things. And then you will find that there's some difference from the template that you use, the human reference, and the reads that you have. So it can be an A here that is different from the whole thing. Okay. So as Arne was mentioning, there are kind of two kinds of mutations that we are kind of interested in. They are the ones that they are inherited, germline mutations, like the Lifrau mini that he was mentioning, the P53. These are usually coming in families, and there's a lot of interesting things, or retinoblastoma. The ones that I'm going to mention here today is going to be somatic mutations. So they are the ones that they are happening in your lifetime. So, and then for looking, and there are several things that the strategy for identifying ones and the other ones is very different. So, and then if someone is interested in the germline thing, we can discuss it later. So you take normal DNA. It can be from blood, for example. You have a tumor, some blood. You sequence, you do the alignment. And then what you do is just compare the two. So for example, if there's some mutation here that appears in this alignment and doesn't appear there, you can make a call that this is a somatic mutation that happens here. Okay? Is there any question about it? It's pretty straightforward. Okay? So there are different kinds of mutations, and Arne was mentioning some of those. The first ones is these point mutations, or substitutions. So this is a change or a base to another base. Okay, so it's very simple. So there are several possibilities, and then you can count the different possibilities. And because it's paired, you have to divide by 2. So you have point mutations. These are the typical ones. There are also something that is important. We are not going to do in the exercise, and don't go into that unless you get a headache. They are called indels, and these are small deletions or insertions, insertion, and deletions. And there are small deletions or insertion. It can be a few bases. So it can be that the polymerase is going and skip one base, and then it's doing these things. But the effect can be devastating because it can change the frame shift. And from a protein that you have at some point, you can get a completely new one, where half of the protein looks like the previous one and the other one it doesn't. Or it can be introducing a stop codon that we will see in some of the examples. It's introducing a point, there's a stop codon, and then inactivating the mutation. So that's a very common one. So the other ones that Arnie also was mentioning is that we see that there is loss or gain of chromosomal material. So in some places of the genome, you can get some places that is duplicated. So you get 2 copies of the same thing. Because we align into a haploid genome here to a genome here, what you will see is that if in the tumor there are 2 copies, you will see that there's more material coming from a particular region because you align this more material into only one place, and that is in the reference genome. So, so these kind of events, they are usually called gains. Okay, there is kind of, you can have, for example, a triplication of a chromosome or a deletion. So it can be, for example, that there is a region, like in the tumor suppressors that Arnie was mentioning, that is lost. So you can see that, for example, when you're mapping, you see that there is no material coming from a particular region in the genome. And this is, it can be, if there is no material at all, it's called homozygous because you just can lose the 2 copies of the chromosome, or hemizygous if it's just lose half of it. So these are called usually copy number alterations, okay? And you have these things. Another type of events that that Arnie mentioned, so for example in MYC, they are going to be very important. This is kind of these translocations, where you can have 2 different places in the genome that rearrange, and a chromosome 1, it becomes with chromosome 10, and they join and create something new. And then you can have part of the read, part of the information is coming from a particular place of the genome, and another part is from the different things. So that happens in many tumors, in particular in pediatric cancers. It is kind of a very common one. In leukemias, for example, I mean, usually they don't have any mutations, so we have many cases of, I mean, babies that they get, and then they get a leukemia immediately, and they have only one mutation, and it's this type of mutations. They are usually very penetrant and very rare events. Yes. Oh, sorry.
Is there a reason that pediatric cases tend to get gene fusions?
So I think my way of thinking is following some of the lines that Arnie was mentioning. I think mutations, somatic mutations accumulate with time. And then in pediatric cases, and there is no enough time to accumulate many of these mutations. So I think there are several routes there. But in these pediatric cases, these rearrangements, they are very rare. They could happen. They are very rare cancers. And they could happen, and they are usually one mutation is enough to create this cancer. So they are very rare but highly penetrant mutations.
Sorry, I don't see why that's still more likely to happen in a pediatric case than a...
No, so what I'm saying, it happens in adults also, okay. The other thing. So in adults, there are several mechanisms that can happen. Just normal accumulation of mutations and rearrangements. In kids, I mean, there is no usually enough time to accumulate mutations to create that through the second mechanism.
I wonder how many pediatric tumors are in cell types that don't exist in adults. We know that the properties of these tumors, for instance, T-cell leukemias in kids go to the brain, but T-cell leukemias in adults don't. So they're really different types of cells, and as we go through life, we transition through different cell types that are specialized for either early in development or whatever. And so when a translocation occurs in an adult and it's not in the right cell type, it might be ignored and you never get it reported.
So I mean, I think you were studying ependymomas, isn't it? So I think ependymomas is one of these cases. There are 3 mutations, if I remember correctly. I mean, I remember discussing this thing with Pablo and other people. There are 3 mutations. I think there is a subtype of ependymoma associated with RELA fusions, isn't it? Yeah, so there is a subtype, and the other ones we don't know exactly what is the cause, isn't it?
There's another gene fusion as well. Yeah, get one.
I see.
Yeah, but yeah, I didn't know why, if there was like a specific reason why this gene fusion
No, I think it's basically these things. They are very highly penetrant mutations. You can find in gene fusions. We have it in glioblastoma. I'm going to mention these things, and these are usually old people, but they are very rare. So because they are other mechanisms. I mean, you have enough mutations to go through kind of more kind of mechanisms that are these things. But fusions happen. I mean, in glioblastoma, there is, I would say, like 5% of these cases that they have very high penetrant mutations.
Are there point mutations that are that penetrant?
Yeah, I mean, usually, I mean, in the classic, for example, many pediatric leukemias, they have BRAF mutations. In pediatric, for example, in pediatric gliomas, they are BRAF mutations. They are kind of the hotspot, the V600. I mean, some of the classic things happened, but yeah, not so many. I mean, I would say. So I was here, and then one thing that's very interesting, and Arnie was mentioning, and also I mean in our talks yesterday, is that because we get a lot of material, we can just see what is in the, I mean, what is not aligned to the human genome. So the strategy is just you take the human genome, you just try to see how the genomic material you collected resembles the human genome, and then you can start making copies, I mean, making calls about all these particular alterations. But there's a bag of things that they are not mapping into the human genome. Also, we saw in kind of in the RNA sequencing in the first one. So, and then you can try to see if there is something else there. So, as Arnie was mentioning, I mean, there are many 20% of the tumors that there in the world, they're associated with viruses. So you can just try to see, for example, there are some virus pathogens. I mean, the last one, I mean, in 2007, I think was this, there was this paper published where they found a very rare type of skin cancer, the Merkel cell carcinoma, and they have a piece of a virus that was incorporated into the genome, and they found it like this exogenous type of material that happens there. We know many tumors, many lymphomas, for example, they're associated with viruses. The role is still kind of unclear in many of those. But this is kind of a fascinating topic. Yeah.
So are you focusing this largely on DNA sequencing or RNA sequencing?
So what we are going to do today, we are doing everything, but it's DNA. Okay, DNA because I think it's much easier to interpret.
So, you know, if I'm sequencing viral sequences, it's because they've been retrovirally.
Oh, no, sorry. In these ones, I mean, you can do, for pathogen identification, for these things is DNA, mostly. But for these things, you can do both RNA and DNA. And usually, in many of the pipeline that we have at the moment for looking at these things is mostly RNA for these things. Yeah. So here is kind of going back to our cartoon. I mean, obviously, you don't see this picture. You have to do an inference of the picture, given that you have these numbers. So you have the frequency of different mutations, the blue, the red, the green, and the yellow. Before these things, this is by definition, we are looking at somatic mutations, so they are all going to be 0 here. Okay. And then what is the frequency of cells that they have this? The blue mutation is now in 100% of cells, the red in 100%, the green in 50%, yellow 25%. And then after therapy, for example, the green disappears and the yellow becomes also dominant one. So the question here is just, I give you a vector of these frequencies of different genes, and can you reproduce what has been the evolutionary process here with this one? The solution is in general not unique, but I think you can get very good approximations in many cases. I'm not going to go into these things, but here is some real data. This is from the Cancer Genome Atlas, and this is some of the data you are going to do. I just took the essence of the columns. I mean, you are going to see in problem 1 that there are many columns. I mean, like this is like crazy. So, but this is the type of data that you can get from one of these cross-sectional studies. And here we can just go through some of the favorite genes that Arnie was mentioning here. So the first column is telling us the patient. Okay, as you probably know, we cannot report the name of the patient in many cases. We'll just put only just a number here that is referring to this thing. So if there's the same number, I don't know here if there's some numbers here that they are the same. That means that there are mutations coming from the same patient. So they are both mutations or several mutations. Each of those is a different mutation. Okay. The next thing that you see is the position in the genome. So for example, the first one here on top, I don't know if you can see that very well. I mean, I can make it bigger. So it's in chromosome 17 in position 7,518,257. Okay, some particular position. And then we should expect a G. Okay, the reference was a G. And however, we found an A. Okay, this happens to be in a gene that is called TP53. Okay, and if this is translated into a protein, that will change an amino acid P, 250 to L. This is the position there. Okay. So these are kind of where the mutations are. And there are all these numbers here, that these are the numbers that I was mentioning, the variable allele frequency. So it's counting how many alleles do you have of this particular mutation. Yes?
For these sort of databases, is the reference always matched, or is it some?
No, so here is basically the protocol that I was mentioning. So you take a reference. I mean, I don't remember what
It's
was
not
there.
necessarily matched.
No, it's not. So, but you match the normal. So you just take, because we're looking at somatic mutations. So you just take your DNA from the tumor, break it, sequence, align to a genome. You just go to the normal. So you do align to the same reference genome, and then you just look at differences. We are looking at things that are somatic. The reference genome, in principle, it should be not an important issue, although sometimes it could be, if they are not. Okay. So, so these are different mutations that happen here. And sometimes they have different, I mean, in some cases, different positions because they can be two transcripts. I mean, sometimes for the same DNA mutation, depends on how, what is the RNA that you produce, you can have different mutations in the RNA and in the protein. So these are the proteins, these are the amino acids. When this is star here, that means stop codon. So it's introducing some mutation that introduces stop codon at some point, and then these things. So these numbers are very important. They are these 6 numbers here. Okay, I'm going to look only, let's look only at these ones here, this one, and this one, okay? These are the frequency of alleles. So here, allele means that we have in most of the chromosomes, not in the X and Y, but we have 2 copies. So if there's a mutation happens in one of the copies, and it happens in 100% of the cells, 50% of the alleles will have the mutation. If what you're sequencing is a tumor, but there is also some cells that are around, because that's happened very often in many of these tumors, then this 50%, it could be a little bit lower. If there is something that happens, like what Arnie was mentioning, that there is a loss of the other allele, so it can be a copy number alteration that delete the other allele, this number can go beyond 50%, and it can go close to 100%. Okay? If there is an amplification, and this happens in also some oncogenes, there's a mutation that gets amplified, and then you have 2 copies, and then this copy with the mutation gets amplified, these numbers can be much higher. Okay? So let's take, for example, the first mutation here. The tumor allele frequency is 98%. So these are numbers between 0 and 100. In the normal is 0. So all these ones, they are going to be 0 because we are looking at somatic mutations. Okay? So we are not looking at germline. So this is chromosome 17, a mutation that happens in this particular gene, P53, in 98% of the alleles have it. Okay? So how is 98% of the alleles? We know that we have, in general, 2 copies of chromosome 17. And what do you think is happening here?
I'm sorry, I'm
Yes.
confused about one thing. I take a tumor sample that contains both cancerous cells and
Correct.
stroma and God knows what all.
Yes.
You can't separate those things out.
You can't. You can't.
So how the hell can you get 198%?
Because these tumors have been selected to be very high tumor content.
Okay.
So
So these are tumors where
it has been,
scaffolding
in general, for these protocols and in this consortia to be in. So there's a pathologist that is going there.
Somebody goes and
They look at,
dissects out.
dissect,
Ah.
and they are making sure that it's happening. Said that, this process is suboptimal. And there are many tumors that we know that the tumor content is very low. I mean, in pancreatic tumors, for example, that we have these issues in many of these things, they have tumor content that is very low. I mean, but in these ones, I'm taking the easiest one. Okay, here we don't want to complicate the whole thing. So I'm just taking tumors where we suspect the tumor content is very high from other kind of reasons. And here what we have is that 98% of the alleles in P53 have this particular mutation. So why do you think? I mean, if a mutation happens in heterozygous, we expect 50%. So 98%? Hmm? Yeah. So okay. So what you have is that there is a copy number that deletes the wild type. And what you have is that this particular mutant goes almost to 100% of the alleles. And this is very common effect. This is a very simple method, for example, to identify tumor suppressor genes. So here we are going to see several of these guys here in this list. They are going to have the same phenotype. Okay?
But couldn't I also say I have 2 copies and both of them have to be defective, as it were, in order for...
That's right. Yeah.
Yeah, I mean,
That's right. Yeah. So I mean, from this data, I don't know that. What I know is that basically there are two copies. One disappears, and the other one has a particular mutation whose effect I don't know. So, and then the biologist has to do some experiment in a cell line or something, introducing the mutation, and characterize the mutation. And this one. So, so this is, I mean, these are some of statistics. I mean, because you don't know the frequency, you have to do some confidence and all these different things. And there are some numbers here. And this is the number of the depth. The depth is something that was mentioned in the first. Is the number of reads that you have in the particular thing that they are basically capturing the mutation, this particular loci or locus in this one. So there are 241 reads that are capturing this particular thing. Okay? So this one. So from this thing here, we expect that this is going to be an important gene. We don't know exactly the function of that particular gene. Yes?
Just to be clear, these are all point mutations, or could they be like next to each other?
Yeah, so there could be sometimes, sometimes you find that there are two mutations going together, and it's very interesting. I mean, you can see that these dinucleotide mutations going together. These ones, I think we were selecting for the ones that they were just substitutions. Here is, for example, an indel in another, and also in this gene. Curiously, it's exactly the same thing. Exactly. So, but most of them, I mean, here is another indel in another gene that is called NF1. That is also kind of important. Yes.
So, is every one of these a different case, or is it?
Is a different one?
Is every single person? Is that every row a different person, or are there duplicates?
No, every row is a different mutation, and they can be. I mean, every mutation. So this is the top of a list. I mean, I just ranked here by the tumor allele frequency. This number is going from top to bottom here. So this is kind of the top of the iceberg of many different mutations. And the idea here, I think in this analysis there were like 200 glioblastoma patients, so a room bigger than this one. We sequence all of those, and then we just look at the point mutations, and I'm just ranking, just taking, there is 100 for each one, so if you have 100 times 100, around 10,000 mutations, and then you just look at the mutations that happened.
Sorry, I'm not sure I still follow. So is it that, would you get someone twice in this list?
You don't get the same mutation twice. You can get different mutations. You can have a mutation in P10 in the same patient. I don't know here if you can see someone that is exactly the same. No, here, for example, here you can see this number here.
Two mutations.
Yeah, so this is the same number here. So that means that it's the same patient. So this patient has mutations in PTGFRA and another mutation in a different place in the same gene.
Okay.
So this one, he has two mutations in the same gene, and it's coming from this patient. And if you are going down, probably you find another times here. For example, there's another one here. This patient here has two mutations, one in P53 and stop codon in 196, and another one in another gene, ATRX, that we know that is kind of important for these ones. Yes?
So you're using an intermediate reference genome.
Yes.
But I notice all of these have normal frequency at 0.
That's right.
Could it be the inverse of that, where like the tumor has the absence of something that's in the reference genome? Like, is that also included in the?
So if there's a deletion, so if there's, for example, a place in the genome that the tumor doesn't have, okay, and in the normal, this is not the analysis that you will do. So you just look at the material, and then you just look at places where you don't have enough material to cover this region, and then you have to do some statistics to try to see that you are confident that in this region there was a deletion. I mean, you can do the opposite. So, for example, what you are saying. So you can take and say, what happens if you just take the normal and subtract the tumor?
Yes.
And you can get some funny things. For example, these ones will appear in this case.
I mean,
Yeah.
is it possible to use like the normal as your
Yeah.
reference? Is there sufficient depth in this?
No, in general, not. I mean, these are usually exome data. I mean, most of the things in TCGA is exome data. I don't know if you're familiar with, I mean, reassemble the whole genome is kind of a complicated thing. So usually there are several strategies, and you have to, I mean, people do it, and now there's a lot of whole genomes and all these things, and especially for complicated effects. I'm going to mention some of those that is kind of scrambling the whole genome and rearrangement again. So reference-based methods didn't work. So you have to go to kind of methods that they are not based on reference. Said that, I mean, kind of this is the first look and the easiest thing that you can do. Okay, there's, as I was saying, what is fascinating about these things is that every day, I mean, we can find kind of a new type of event that we were not imagining, and then you can just go there and doing the whole thing. So the next one, for example, this is another very interesting example. Is in in this patient, 06, 07, blah, blah, chromosome 7, a C to a T is not in the normal, so we know it's somatic. It's happening in this gene, EGFR, and this is a synonymous mutation. It's interesting. This is a synonymous mutation, and it happens in 97%. And one of the things here that we see is that the number of reads that are covered in this region is very, very high. So there is a lot of material there. And 96% or 97% of these reads, they have the particular mutation that happens there. Okay? This is just compared to what is in the normal that is supposed to be only like around 100 or something like that. So you should expect 100 reads, but you get 2,000 reads that are in this region. And of those, 97%, they have this particular mutation. What do you think is happening?
Excuse me, are these mutations filtered for cluster events?
For? No, this is not filtered. I just selected kind of the top of the most common with loss of heterozygosity.
Because sometimes one of the problems is that there is these events, there is the clustering events.
Yeah.
Is due to a sort of mismatch of the
Yeah, exactly. So I'm going to mention here about the problems of, and I think this is why it's an interesting problem, because the statistics are non-trivial here. What?
Especially for those genes that contains the same mutation.
Exactly. EGFR, for example, is one of my favorite genes in glioblastomas. They are super rearranged. The whole region is highly scrambled. And what is happening here, I can just give you the solution. This is a region that is very well known in glioblastoma that is highly amplified. It's a region in chromosome 7. It's one of the marks of glioblastomas. There is a lot of copies around the EGFR gene, many, many copies. It could be, I will expect here, this would be like if in the normal this 123, and you should expect 2 copies here, you should expect here, 40 times bigger or something like that. So this region is heavily amplified, and this mutation that was there, it appears to trouble with the whole thing. So this is one clear identification of, for example, this is an oncogene type of things, where you see the mutation that is heavily amplified in some particular region. The next one, for example, is another one that has this ATRX mutation, and this is a mutation also in 96% of the cases. This happens in the chromosome X, so then you have to try to see if this is a male or a female, because if it's happening in the chromosome X and it's a male, then you can get numbers that is close to 100%, and so on. So, it's very interesting. So retinoblastoma, for example, the story that Arnie was saying, RB1 here is the retinoblastoma gene. In this patient, in patient whatever here, 26 something, there is a mutation that introduced an arginine here, put in stop codon here in this particular position. And then you can just try to see if there's a loss of the other one and trying to infer what was going on. But having a stop codon is clearly some of the marks that it can be, you suspect that it can be a loss of function in this particular one, and you suspect that this can be a tumor suppressor in this one. The same thing, for example, there's another stop codon, and you suspect that this gene, P10, is another gene that is going to be, has mutations that are loss of function, and probably is a tumor suppressor. P53, another one that has the same thing. This has a different mutation, and this is not, but retinoblastoma is another mutation here. So this is kind of the type of things. I've been selected here some of the genes. When you work in the data set, you are going to see that it's very messy, it's very different. But these are the things. And obviously, if you want to say, well, if now you want to study what are the genes that are important in this list, some of those probably they are not important, some of those they are going to be important. Typically, you get like 100 mutations for each patient. The fact that you find so many times the P53 or EGFR or PDGFRA and some of these ones, they suggest and you suspect that there is something that is selecting for these mutations. And there is, as Arnie was mentioned, the mutations, they are going to be not uniform or random, but there is selection there that is doing. So we suspect that there is something important, and then you can start focusing on these particular things. So this we identified many different genes. Okay. This is, for example, how this variant allele frequency looks. These are, I think, a pediatric leukemia. Before therapy and after therapy, you just put the variant allele frequency, so the first number. So there we sequence 3 genomes. These are whole genomes. You sequence the normal, so we look at somatic mutations. We subtract all the normals before therapy and after therapy. And then you just put the variant allele frequency here, the variant allele frequency here of the different. Each of those is a different mutation. And here is a nice example where you can see here that most of the mutations, many of the mutations, they are here. They are going to be in common between before therapy and after therapy, and they are around 50%. These are tumors that they have very high tumor content. There are some mutations that happen to be in a region where there is loss of heterozygosity. And some of these mutations, we know these ones, they're in a region where there's a loss of a chromosome that they happen. So the frequencies, they rise to 100% instead of 50%. But what is very interesting is that there are mutations that are specific to diagnosis. So mutations that happened before the treatment was given to this particular tumor. And there are mutations that happened only after, in the relapse, after the treatment. So if you are trying to look for mutations, for example, that they are associated with resistance to the therapy, you probably will try to see if there's something here interesting, I mean, in this particular cloud of things. You can see that there's a subclonal population here. I mean, there's a lot of structure here. And reconstructing all these different histories from these pictures is pretty interesting. So as I was mentioning, gene fusions are very important in many tumors. In the problem set, we are not going to touch gene fusions because that's a complete different beast. But the typical idea is that you look at your reads, and then you look at part of your genomic material that is mapped into a region of the genome, and another part that is mapped to a different region of the genome. There are these kind of events. So most of the protocols for sequencing, what you do is just you fractionate the DNA, and then you sequence in both sides. So this is like a few, let's say 1,000 bases, and you sequence from one side 100 and another one from 100. So you know that these two pieces, they are kind of contiguous, but when you just map, this is mapping in particular region in this particular gene, and this one is mapping to a different gene. Usually we do it in both with DNA and RNA in these cases. And the other kind of evidence is these ones. These are reads spanning diffusion, where you have half of your information is mapping to a part of the genome and the other one. And from this type of information, we can reconstruct very accurately what is the breakpoint where these things happened. So this is, for example, this is one of the examples that Arnie probably can mention these ones. These are two common, this is a very common rearrangement that happen in CML, in some leukemias. And these are chromosome 9 and chromosome 22. They break, they exchange, and they create a new gene that was not there before, BCR-ABL. And this is a very interesting gene. This is one of the examples of these kind of targeted therapies that is kind of the standard one. And this is called the Philadelphia chromosome. This is one of the standards. In leukemias, they are very often, and we found many. In gliomas, for example, if you go to gliomas, you can start looking at this one. We found a few years ago one of the first fusions there, and it happens in chromosome 4 between two different genes, FGFR3 and TAC3. And these are, again, they're events that they're super highly penetrant. So you have the fusion, you just clone it, put it in cells, and they start proliferating very fast. And usually these genes, they don't have many other mutations. So they have only like kind of a fusion that is a very rare event that happens there. But what is interesting is that there can be some particular drugs that they are targeting some of the protein products of this fusion. And then there has been a lot of development in this particular area. And then microbial detection in cancer is something that is super interesting also. I mean, it's one of my favorite topics because there is a lot of interplay there that is not very well elucidated. And the idea is that you just look at exogenous material in your sample. If you have whole DNA, so you can just look at DNA that is coming from different things. If you have RNA, you can look at transcripts or some particular RNA material that is there. So this is one case that usually Arnie, I was expecting Arnie to give an introduction on this, the Burkitt lymphomas, because it's one of the classics. So this is, these are, for example, endemic Burkitt. This is, I mean, Arnie, you can tell the story probably much better. But these are cases that happened in the equatorial belt in Africa. It happens in kids. They have these kind of very nasty lymphomas. If you go to the Wikipedia, you can see these pictures that are very distressing. And then, so, and they're associated, and this is known, with Epstein-Barr virus. In this, the Epstein-Barr virus was identified through samples in this particular one. And then they are usually kids from 4 to 7 years old, more in boys than girls, and it's unclear why. And it's also unclear why it's happening only in this part of Africa. I mean, why only in these particular regions. Burkitt lymphomas, these are B-cell lymphomas. They happened also in the West, so they happen here, but they are very rare, and they're usually sporadic in adults. So it's a very, very different structure. So we compare the different mutations that happened in endemic, and we compare with some of the sporadic cases that happens here in the West. These are from the US. And then we look at the presence of different viruses. And here, for example, Epstein-Barr virus, it happens in 100% of the endemics. and it happens only in 4 of these 20 or something that happens here. We found also cytomegalovirus in some of these cases. In the African cases, these are kids coming from Uganda. Some of those, Kaposi sarcoma, that was another of the viruses that was mentioned yesterday, and some other viruses that was kind of interesting here. One of the things also that is interesting are the mutations here. These are some of the mutations. So each column is a different patient. So each column here is a kid coming from this Uganda trial. And these ones, they are adults coming from the US. And when there's a color thing, that means that there's a mutation in MYC. It was one of the genes that Arnie was mentioning. ID3 and other genes, P53. And then one of the things that we were trying to do is what are the mutations different between the cases in the African one versus the ones that are here. And there are several differences there. There's a very interesting story about it.
Can I add a few things?
Yes,
Okay.
definitely. Yes, of course.
So, Burkitt lymphoma was actually the first virus that was proven to cause cancer in humans. And it was this relationship of finding Burkitt lymphoma in Africa. And these are B-cell tumors. You'll learn tomorrow in immunology. Those are the cells that are the precursors to make antibodies. This distribution in Africa is the distribution of the malarial belt. So that's the first thing to remember about this. I'll get back to it in a second. The virus, Burkitt lymphoma, is found in about 85–90% of people in the United States. So that scared a few people when that was found out. And then it was shown to cause infectious mononucleosis in people in the United States. So here's a virus that's associated with a malignant tumor in Africa that is associated with Burkitt's lymphoma. Burkitt with it's associated with infectious mononucleosis in the United States. Now, let me just describe infectious mononucleosis. It's called the kissing disease because you transmit it through saliva or kissing. The virus replicates first in your nasopharynx. It's an epithelial cell virus replication. And then it moves into the B cells of the body. However, it's not diagnosed by B cells. It's actually diagnosed by another type of lymphocyte that attacks the B cells. So the B cells are seen by virtue of the fact that they have Burkitt lymphoma in them as foreign. And the T cells come along and they kill the B cells. And the T cells look very abnormal as they're doing that killing. So the diagnosis is done by blood sample and as an abnormal T cell, even though the T cells are actually defending this. Right? Burkitt lymphoma, though, so that's pretty interesting. And then back to Africa. This is the malarial belt, and malaria is immunosuppressive. So these kids that have it would have infectious mononucleosis and have a B-cell infection, but for the fact that their T cells are no longer killing them, right? So for example, if I was to take blood from everyone in the room and we just culture it, B cells and T cells would die off. But if I was to get rid of your T cells, I would find 1 in 1,000 or 1 in 10,000 of your cells has Epstein-Barr virus in it. It's a latent herpes virus, latent in the B cells, and it's being kept under control in your bodies by your T cells. Right? And those cells are immortalized. They will grow forever, being a cancer cell, therefore. So we are all carrying these cells, and our T cells are protecting us. The people in the United States that actually get Burkitt lymphoma are often the immunosuppressed people. Right? Now, that sounds like I could explain everything until I now mention the last thing. This virus is associated very closely, probably causative, with nasopharyngeal carcinoma in China. No place else, just China. A little bit in parts of Africa, but not overlapping the belt yet. Nasopharyngeal carcinoma makes some sense. Remember, it's the kissing disease. It replicates in the nasopharynx. So that's the first place. Why it's causing that tumor, we have no idea in China. Just no idea. In fact, nasopharyngeal carcinoma exists in the United States in the absence of this virus. So it's spontaneously mutations in the United States, it's the Virus Association. The final thing that was done was that they thought that it was genetically predetermined in Asians. And so the Asians in San Francisco were in fact studied for 3 generations to ask the question, did they have a very high rate of nasopharyngeal carcinoma and associated with the virus? And the answer is it's somewhere between Caucasians and Asians in China. So it's a very unsatisfactory answer. It's partly genetic and partly something else. And the problem is that while the genetics stay the same in these individuals, so do the foods and the other things they eat. Right? So does the environment. They don't really change their environment very much while you're living in San Francisco because there are good Chinese restaurants in San Francisco. So this is really an enigma of a virus, but it's one of the herpes viruses that's latent, and it's a cancer virus, and it's even a cancer virus in us, essentially, but our immune system protects us. This was a clue right from the beginning of what we're going to start to talk about tomorrow, which is how the immune system interfaces with the cancer cells. This is a really good example of that kind of thing.
Yes, I mean, going back to these things, I mean, the other thing that I think is fascinating, and this for me, I mean, first of all, I mean, Arnie was mentioning there's like these 5 or 6 or 10 genes that they are going to be important for cancer. The rest are going to be passengers. Going to be our first question. But they have to be the right 5 and in the right combination in the right time. So now, I mean, so just looking at this pattern here, they look like random things. I mean, you see that mix, they happened in many cases, ID3 and all these things. But I mean, here, I mean, obviously there is no statistical power to try to identify patterns. But in many tumors, we start getting enough power to try to see what are the combinations of mutations that should happen. I mean, and it's interesting. It has to be like 5 mutations, but not 5 random mutations in these genes. It have to be, depends. There are several gangs of genes that they have to do it. And the same thing happens with external factors. I mean, like viruses, for example, we are finding in Hodgkin and in all these cases that tumors that they have the virus, they have a different mutational profile than the tumors that they don't have the virus. So there's an interplay there with the viral world that is, I think, quite interesting between the mutations and the others.
This part, how are these individual cases being divided into endemic versus?
Oh, these ones 2 different cohorts. And these ones coming from, this is, I mean, the story started with this guy. This is a doctor in Siena. That, and then he went to Africa for several years. He was treating these kids, and he was collecting samples.
I mean, Burkitt's lymphoma is it's a diagnosis, it's a disease, it's a description of symptoms, yes?
Yes, that's right. So, I mean, if you just go to Wikipedia, they have these awful pictures. I mean, it's very easy to identify because there's these kids with this jaw there. So he was treating them. He was collecting samples from Uganda. It's very hard to get samples from some places and all these things in this age. So he was collecting these ones. And then there was data from here in the US. And these are patients that are coming from the US. They were collected here in the US.
So they are by definition sporadic.
They are by definition sporadic. Okay. And these are adults and these are kids. So they're different populations. These are kids coming from Uganda, and these are adults coming from the US. So they are 2 different populations. So let's go to our first question here, and this kind of a big question. And I think there are many different ideas in the market, but I think there are many different ways of improving what is going on. So the idea here is the following. So you have this cross-sectional study. So you have 100 or 100. We are going to, the data set that we are going to mention in glioblastoma, there are 400 patients, I think. And then each of those, they have a few hundred mutations. So this is just only the exome. So you have 100, 100, 100, 100, 100. So you have a list of 100 times 400. So 40,000 around mutations. I don't know exactly the list. And then the question here is how you find the ones that are going to be important mutations and the ones that are going to be there. We expect only a small fraction of those to be important. Okay. And so again, so this, as I was saying, every tumor has a different mutational spectrum. So you don't find in these patients too that they have exactly the same 100 mutations. I mean, I found it only once in my history that it was like we were sequencing and the same person was analyzing 2 tumors coming from 2 different places, and they exactly have like 100 mutations, and they were exactly the same. And what happened is the doctor was collecting it. They sent 2 samples to different places from 2 different collaborations, and they the person who was analyzing, he was just looking at the data, and he was exactly looking at this. It's impossible. I mean, how is something like what Steve Goff was mentioning. I mean, that means that there's a transmissible cancer or something. So it was the same cancer found in, but at the end it was the same person that was sequenced by 2 people. So, so how do we separate these things? So the main path that many people are following are these cross-sectional studies, so taking hundreds of patients. And the problem is how you do kind of performance statistics. And this is going to be related to some of the problems that are going to be mentioned later, although I'm very late here. So the first criteria is this convergent evolution, so the idea of recurrence. So like we saw before, if you see that there are 100 mutations, you suspect that if mutations, there are 20,000 genes, and there is 100 mutations, it's very rare to find exactly the same mutation or exactly the same gene mutated in 2 different people. If you find it in 5 people or in 6 people or in 7 people, then you start suspecting that there is something that is non-random there. It could be selection or something. So these are typical things. I'm going back to this plot that Arnie was mentioning. This is the number of somatic mutations per megabase. If you do whole genome or exome, numbers are very similar because this is normalized by the size. What changes that is different is that the power to capture subclonal mutations. So usually when you reduce your target place, your sequencing to a smaller one, you have sometimes, you sequence deeper, and then you can find mutations that happen only in a small fraction of cells. So sometimes in exome, they can be higher rate because you are capturing some subclonals that they don't happen. But these are kind of the numbers, and Arnie was saying some of the tumors that have very few mutations, that they are kind of usually pediatric tumors. Some of those that they are associated to some UV damage or so some cases that they are extreme there, they are usually mismatch repair deficiencies associated. And then the idea here is just you just take all these cancers and then you just try to look at what are the things. If you do, for example, these things with melanoma, that sometimes they have thousands of mutations in the exome, you're going to find a lot of these mutations. They are, it's very hard to do statistics in melanoma, these ones. And these ones are usually the easiest one. So I like very much leukemias and all these things because they're very clean. So there are only a few mutations, and they're usually very clonal, and you find it there. So this is the first example of a leukemia that we found some years ago. This is like in the beginning when we were kind of applying these things. So this is a very type of B-cell leukemia. It's called hairy cell leukemia. They are very rare. And then we start sequencing these tumors and trying to find mutations. So this is kind of a representation of the mutations, and these are called circles plot. This is circles. And then these are different chromosomes, chromosome 1, chromosome 2, and each of these is a different patient. Each of the circles is a different patient. Okay? So there's the blue patient, the pink patient, and these ones. And these leukemias, they have very few mutations in the exome, very few. So it's around 10 to 20 mutations.
The radius is moving me along the chromosome, or what?
The what?
The radius on this plot?
No, the radius is different patients here. Oh, this one here. No. So these are different patients, and the position in the chromosome is here. Okay. So here, for example, I mean, you see these things, and there are different positions. These are different bands. So the mutations, they happened, and this is the typical thing that you find in one of these tumors. This is a very simple one, because you don't need to do any statistics to find what is the culprit here. So you go here, and then you can just count how many times this gene is mutated in different patients. Okay. So just very simple statistics. Just count. And then this is reflected in this histogram here. So this is only 1, 1, 1, 1, 1, 1, and you can go, and then like a delta function in this particular gene. So there is a gene in chromosome 7 that is mutated in all the patients. So we went there and said, well, what is this gene? Well, it results that it's not only the same gene that is mutated, but it's the same position in the gene that is always mutated. It's always this gene. And then, this is a gene that is a gene that Arnie has not mentioned. It's related to RAS. It's called BRAF. And it has mutations. These are mutations also that happens in other tumors. In pediatric gliomas, for example, is a very common one. And this 100% of these tumors, they have a mutation in this one. So they have genes that they have only a few mutations, and they are always happening these things. And now it's used for diagnosis, for example, of this disease. So you have the mutation. You suspect that there is this one. You can look at the mutation. It's there. The interesting thing also from the practical point of view is that BRAF inhibitors, this is a mutation that happens in 50% of the melanomas. And there have been a lot of development trying to look for inhibitors of this particular mutation, not only this particular gene, but this particular mutation. And now there are many clinical trials, right, and they're kind of very interesting things going around. There's a lot of case reports and clinical trials going around on using BRAF inhibitors in hairy cell leukemias. So this is something that you don't have to do a lot of statistics to find what can be an important gene. And we know for sure that this is not the only one. We don't know which of the other ones. We don't have a statistical power because there's only a few of those have been sequenced. But we know probably that this is not sufficient. It's necessary, but not sufficient cause in these things. So how about the tumors that you are going to see this afternoon? So like gliomas. These are like 100 patients, the same type of plot, different chromosomes, chromosome 1, 2, tac, 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 tac. And then here is kind of the distribution. So you're looking at all these things. And then the question here is which of those is going to be the important genes here. So the first thing that you can do is doing the same strategy that you were doing before, counting the number of mutations that happen in different genes. And this is the histogram that you have here. And then you are trying to look for peaks in this histogram. Chromosome 7, there's a peak here. This is EGFR. This is one of the genes that we found, one of the classic glioma genes. You go here, 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 here. Yeah, this chromosome 17 is P53. There's another peak that happens here. And there are other peaks that are around. And then the problem here, it becomes that some of those is, I mean, you know what is the. So here is the number of cases. I think there are 283 gliomas. Just count how many. You just rank the genes by the number of mutations that they have. And then, so the first one is P53, and there was like 30, and then so on, and there's kind of this curve. It's the blue curve. So this is the order by the frequency of a number of cases that they have these particular mutated genes. So axis is genes ranked by the frequency of these things. And you see that there's kind of a peak here that is easy to identify. And there's here a long tail where things that are in around the 2%ers, 3%ers, and all these things that we don't know exactly how, if this is something that is good or is not good, and how to do these things. We know for sure that there is a lot of information here because we know from other tumors and a lot of information. There are good players there, hidden, but we don't know exactly which are the other ones that are going to be important. And I think this is the question. Yeah, sorry, I have these things. We know that there are many of those. So that's the first question. And the problem set, what we do, so Yunfi prepared the data, downloaded the data from TCGA. There are 33 tumor types, 10,000 tumors, they're exome, and then you get the variant file. So this is tables where you have very similar to what I was showing, mutation, 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 and then annotation of the different parameters. There are some annotations that are from the sequencing, some annotations from previous information, if this is something that is commonly found in other things and other things. You don't pay attention to the rest of the columns because it's, I mean, you can just spend your whole life there. But, yes?
In the example you just gave, there were two genes which were fairly common, and so those two are associated with the tumor. And then there's a lot of other genes that are not so common. If you build up the statistics of that, do the other genes fall into different kinds of tumors, subsets of tumors,
yes.
types, tumor types? In other words, the other genes aren't irrelevant in ways.
They are not. So for example, I was mentioning just counting the mutations in a gene.
Yeah.
So the exercise that I'm going to give there is going the mutations in the same position in the gene. So that's P53 mutations. They are hotspots, and you're going to find hopefully some of these hotspots. So there are some places that they have more mutations, but they happen kind of distributed everywhere. Other genes, so that the gene that you are going to find soon, it happens the mutations always in the same position. So and this is very interesting because it's a hotspot. It's always happening the same mutation in the same position, and this is something that we know is well characterized. There are other strategies, for example, inactivating mutations. If you have an enrichment of inactivating mutations in these genes, then you can just increase your statistical power to start fishing these things. But you have to go with some prior knowledge of what is there to start fishing some of these.
I mean, the pathologist said there were 5 kinds of tumors.
Yeah.
The goal would be to take them and subset them into the other kinds.
I would say the other way around. So pathology, and this is kind of interesting because, so in pathology, I mean, people are looking at the shapes of different cells and then making a call about different subtypes. What is changing now in this era of sequencing is that now from molecular data, from expression, mutations, and all these things, we can have classifications. It's, in many cases, they don't match, I mean, in the whole thing. I mean, for example, in gliomas, there is no subtype except for IDH1 mutants that they have. That they're a very particular kind of tumors. They have a particular methylation profile, and they have a survival, so they are doing much better on these ones, and they are associated with a particular kind of a structure. So, for example, the fusions that I was mentioning, these rare fusions that happen in gliomas, they're interesting. They are kind of the only defining things. They happen in different places in the brain. They are happening close to the boundary of these things, and they have particular type of things. I mean, pathology is usually they take in consideration the spatial thing at the lower resolution, but not at this. So I think now it's very interesting because now many of the pathology classification is being revisited because of this particular issue. Okay, so I have to go a little bit faster because if not, this thing. So the next thing is reconstructing the evolutionary history of tumors in longitudinal cancer genomic studies. So now what we have is not just we have a snapshot of what is the population, but we can have in some of these patients, we have information that is longitudinal. And then, so I'm going to give this example, and this is the example that we have in problem 2, that is glioblastoma. It's the most common type of brain tumors in adults. It's around 10,000 cases in the US every year, and the survival rate is not very high. It's usually diagnosed because a person starts having some symptoms, clinical symptoms, taking a picture of the brain, an MRI, and then they see that it's a mass, and usually in the next couple of days, the surgeon removing the mass and giving some therapy that I'm not going to the details. So this is a mutation generating therapy. It's called temozolomide. It's this agent, and radiotherapy to these ones. And this is prolonging the lifetime of the person from the median lifetime from, I don't know, a year to 14 months, although there are some patients that do a little bit better. So, we try to understand what is exactly happening and how these tumors evolve the therapy, and what is the initiation of the tumor, how these things work, and all these things. Again, I'm not going to go in details. So the data that you are going to have is data from like 100 tumors. I believe they are like around 100. We already subtract the normal, so the mutations that you are getting there, the somatic. You don't have to worry about these particular things. And then will be mutations in diagnosis and relapse. And then the thing, I'm going to ask you to look for some particular genes in TCGA, that they are going to be the hotspot mutations in TCGA. I'm going to write these things right away. Yeah. So go to TCGA, GBM. This is one of there are 33 files, I believe, in this folder. There is one that is going to be GBM. There are data from 400 patients. And look at the hotspot mutations here. So that means that this is happening in the same nucleotide position in patients. That happens in more than 6 patients. 6 patients, okay? It's just a counting exercise, okay? Very simple. So you can take the table, and then even with Excel, you can do it very, very quickly. You don't do anything. For the ones that they want to do a little bit better, just do the analysis that Arnie was mentioning. Just take all the tumor types. Take the 33 TCGA, and then look at all the hotspots here. Just rank the most common mutations in cancer. Okay? So you have 33 files. So you can just concatenate them, and then trying to see how you look for these particular hotspots. Okay? This is only for if you want to spend more time. I mean, due to the limitations in time, this thing. So this is problem 1A, problem 1B. And then you should be able to generate a figure very similar to what Arnie, this kind of Manhattan plot, that they are taking some common genes and how frequently they happen. Look at hotspots because they are easier to do than counting some of the mutations. Okay? So now with these genes that you are going to do, you are going to take longitudinal data, GBM. So that means that this is before treatment, so diagnosis. There is treatment. And then you have relapse here, okay? And then just look at these mutations that you find. I can tell you, for example, P53 is going to be there, several mutations in P53. What's happened with these mutations in P53 along the evolution? Do they appear before the treatment? Do they disappear in the treatment? Are they conserved in the treatment? And there are other mutations that appear here, okay? So a summary usually we have, this is for this data. So the data was collected by one of our collaborations in a paper in 2016. So yeah, don't look at the paper first. I mean, just try to do it yourself. So, but these are some of the genes that appear more mutated in these tumors. And then you can look at each of those. Each of the nodes is a different gene. The size of the nodes is the number of mutations that they have. So P53, for example, is very common. P10 is common, and F1 is common. And the arrow means a patient or a set of patients where we know that the mutation happens before in one and in the other one. So if it's a mutation that happens in the diagnosis and relapse, we know that it's a mutation that probably happens in the very beginning of tumor evolution. If it's a mutation that doesn't appear at the diagnosis but appear at relapse, we know that it's something that is late. So we can start ordering these things, and then you get these kind of networks where you have very early events in these things and very late events happening. Okay? So I'm not going to the same thing. I mean, this in leukemias is great. So we have many papers in leukemias because you can longitudinally sample many of those chronic leukemias. You can just do at different time points, and you can see, for example, what is the pattern of how the mutations appear, disappear, and I mean, you can really do great things. The other thing that you can start doing, as I was mentioning, is that these are different mutations in leukemias. This is in CLL. And you can try to see if there is a particular structure in the mutations. And there are some mutations that they like to be together, or they don't. And they usually inform certain things. Okay, I have only like 5 minutes. So the next thing is this problem of, and I think it's fascinating, this archaeology of human cancers. And the problem is the following. So we know that mutations there, although, I mean, the mantra in biology, the mutations are random. What do we mean by random? I mean, it's not a uniform distribution in the genome. So mutations are due to different processes. And different processes, they have different biases. And we know that in cancer, as Arnie was mentioning, different chemical compounds introduce mutations. But these mutations, they happen in particular places. So, and we know that there are many factors that are going to be important. As Arnie was mentioning, the amination of Cs, I mean, they can give you U's. When this is methylated, so when there's the methyl here, the deamination, they give you a T that is much harder to repair. So the aminations of methyl 5-methylcytosine, they give you dithiamine that is much harder to repair. And this is happening in every cell. This is a spontaneous deamination. So it's a process that is happening. Because, yeah, this is. And this is happening in the background. The methylation happens in the background that is a C followed by a G. And this is something that you are going to find. So there.
The methylation thing, it doesn't interfere with the phosphate backbone or anything.
Not at all. It's just in the base.
Yeah, so here, it's just in the base. Here, so.
Well, I mean, and it doesn't interfere with the ability of the bases
You make base pairing fine.
Yeah. Yeah, so it's basically this. It's adding this kind of methyl group there.
Instead of pairing with a G, it pairs now with an A, right?
Right.
So that's the mutation ultimately that you get.
So
Is GC to.
So as Arnie was mentioning, this is fascinating. I mean, this is one of my favorite topics because, I mean, we know that there are certain things that we, some processes that we know very well. As Arnie was saying, defects in DNA repair, that they are very important. Arnie mentioned mismatch repair in some of the colorectal cancers. So you have tons of mutations that happen there in these tumors. And they have a particular flavor. Also, they happened with a particular flavor. BRCA mutations, they happened in uterine, ovarian, sorry, and breast. And that's a very common mutation. The mechanism, exactly how these are related to cancers, is still.
So this is pretty strange stuff if you think about it. So BRCA and mismatch repair are both DNA repair processes, right? And DNA repair processes could happen anywhere in your body. So you'd think that your DNA repair mechanisms would be all throughout your body. And there's evidence for that. Yet the cancers you get are cell tissue type specific. BRCA is ovarian and breast. Mismatch repair is colorectal and pancreas. And, I mean, so the mismatch repair defects are also, like tumor suppressors, are also very tissue specific in their effects, when in fact they should be repairing DNA all over the body. So those are problems, if you're thinking of problems to think about, those are problems we just don't understand.
Yeah.
What gives rise to this, the tissue specificity is functional specificity for the cancer.
Yeah. So they're exogenous factors. So they are things, I mean, these are things that we live. The other thing that Arnie was mentioning is I think there's a difference in different tissues, but also I think in different people. So I think the effectiveness of repair, I mean, we see, for example, I mean, when we get certain things, I mean, it's different in different people. So I found that also kind of interesting. Exogenous factors. So pyrimidine dimers, mutations in UV melanoma. So UV radiation introduces a particular type of mutations in melanomas. And when you sequence melanoma, if you just go to the data set of problem 1, you can go to melanoma. And then you start looking at the mutations. And then you can, doing some small statistics, you can try to find that the mutations, they have a particular flavor there. Food, as Arnie was saying, aflatoxin, for example, in food is one of the things that have been related. Different tobacco compounds. Occupational exposure, there are very interesting things in haloalkanes and different things that people have been working with different compounds and they can get some weird cancers. Arnie was mentioning these chimney, how it's called, chimney boys. The word of this. Yeah.
Chimney sweeps.
Sweeps, exactly. So, this is clearly an occupational exposure. So there are some different compounds in the smog or something there that they are doing these things. And one of those that we are very extensively studying is chemotherapy. So chemotherapies, they introduce mutations in a very high rate. And when you see, for example, tumors that they have exposed to different chemotherapies, you can see different type of mutations that happen. So just to tell you here that when people are saying that mutations are random, what random means here is it's not a uniform distribution. And that's going to be very important to modeling, for example, things about what genes are important or not based on recurrence, because not all the mutations in the genome, they have exactly the same probability of getting mutated.
I can point out one other thing that's useful for the things you're talking about. The mutagens in the environment, carcinogens in the environment, make specific mutations. So that, for example, in China, the peanut crop is often contaminated with a mold called Aspergillus, which makes a toxin called aflatoxin. Aflatoxin causes a mutation in the sequences in the p53 gene at a codon, a specific codon, right, which is arginine, right, to some other amino acid at codon 249. So if you're looking at liver cancers, because this is liver-specific toxin, if you're looking at liver cancers and you see 249, right, you know you've got a carcinogen in the environment. This is like archaeology.
Exactly. No, I think this is archaeology. What is fascinating is that and I'm going to go to the problem here. So the other thing is that at different scales, the ones that I was mentioning is microscales, substitutions, so small changes in bases that we know the preferences. Mesoscale, there was a paper in I think, Science, I'm mentioning here 2 weeks ago. Very fascinating. So they saw that indeed, the DNA, especially when there is replicating, can form these loops. And these loops, they are the substrate for a deaminase enzyme, so ApoBec3A, that is introducing mutations. So the mutations, they happen specifically in some particular places in the genome. And then they create these hotspots. They are places where you see different people, and they have mutations because this particular enzyme recognizes the substrate. And they use this thing to separate something that they are kind of hotspots that they are not really meaningful. It's just because they happen in the background, from something that mutations that they're repeated, but they are not in hotspots of these particular things. So it's a very interesting thing because this is secondary structure in DNA. So you just fold the DNA, and it's not only just the base pair that is happening, but it's how the conformation is happening. This is basically binding to the protein, and they have a very well. This 2 weeks ago, I found this paper when I was preparing the lectures last week, and I went to the room there, and it was just. And the other thing that is super interesting also, this chromatin structure. So we know that the DNA is not a linear piece, it's folded and all these things. They are very interesting ways of folding these things. And when you look at mutations, there are places where chromatin is open and chromatin is closed, and there's different mutation rates, depends on where they are, and different processes. This is one that has been kind of famous in the last few years. This is called the rainfall plot. So this is whole genome, I think it's a breast cancer, I think it's a breast. And then you just compute, so you take the whole genome, you just put it in one line, you just compute the distance to the next mutation, and to the next, and to the next, and to the next, and all these things. So it should be following an exponential distribution. You just plot the log to the distance to the next one, and it follows like that. And there are places in the genome where you have mutations all going to each other. Someone mentioned before, isn't it? So there are mutations that happened in particular locations in the genome, much more frequent than you expect. There is interesting rearrangements here and a lot of story. So the main, stopping probably here. There's some, and we can continue with the single cell. There are many different methods that people. Yeah, we can spend some time. So the idea here is that how can we decompose these different mutations into different kind of directions. So what I would like to do is like taking a tumor, I say from a patient, and I say, well, there are 30 mutations that have been done to aflatoxin, 25 that they are due to tobacco smoke, and these things, and perhaps other compounds that this person has been exposed that is unaware of, or something like that. So a typical technique for doing these things is that this non-negative matrix factorization. The data that you start is many different tumors and substitution patterns. So how this is done is just you take 3 bases. So this is in DNA. So just look in base number 1. Let me just go to the other side here. So you have base number 1. Here you have you fix for these things a pyrimidine, and number 2, because the other one is going to be complementary. So there are 3 possible changes that you can do here, 3 possible changes that you can do here, and 4 possible nucleotides here and possible nucleotides here. So you look at, for example, all the possible mutations, AA. So here, let me just see a C, going to AGA, for example. And then you start counting how many of these mutations do you have. This is 4 times 6 times 4. This is 96. So it's a vector. You classify the mutations into a vector in 96 dimensions. So it's going to be positive numbers. They are going to be counting. Okay. So your data is going to be, and the data that you are going to play here is going to be doing non-negative matrix factorization in 560 tumors, whole genome. So Yunfei already prepared the data. So you have a matrix that is 96 times 560 genomes. And this is going to be the count. Huh?
With hyperspectral analysis.
Yeah. So, and then you have your data. And the strategy that many people are following is that you want to decompose these things into a lower rank representation with positive numbers. And there is a set of techniques here that are looking for kind of decomposing a matrix into a product of 2 positive matrices. So 2 matrices with positive non-negative elements. So it can be zeros or these ones as a product of those. Usually minimizing some type of norm, in this case is the Frobenius, constraints that all the elements should be bigger or equal to 0. Yeah. So, and the idea is at the end, you will find the tumor, and it will be 30 mutations. They're associated with methylation of CpG. So there's some catalogs of these mutations. This is the pioneered and promulgated by the Cancer Institute in the Sanger. And there are different signatures. For example, the signature 1, it happens, these are different trinucleotides, and this is usually the CpG, these things. And they've been looking at different signatures and cataloging signatures in many different tumor types. Okay? So the exercise this afternoon, for the ones that they're interested in this exercise, would be taking these 560 whole exomes with the data already counted and processed by Yunfei, and trying to see what are the different signatures that you can identify in this one. Yeah. There was a paper also last month, that they've been doing this experiment in vivo. So, or sorry, in vitro. In vitro. So they've been doing this experiment in vitro. They took, I remember, there was like 50 different cell lines. They introduced compounds, 79, and then you just propagate. You sequence before, you sequence after, and then you have a biological basis for all these things. So you have each of these peaks correspond to a trinucleotide things. They are going to be a sum of positive numbers. And then you can try to see if this aflatoxin is here, and there are different ones that you can go there. It's a very nice paper. Okay, and then these are...
So the bottom line is this decomposition method actually works, yes?
I will not be so optimistic. So, I mean, it's giving you some...
It's a great idea.
It's a great idea. So I think the idea is great. I mean, so there, so you get a result. What result, how this match to the biology and all these things isn't clear. So what is the best mathematical approach to uncover signatures? The simplest thing is just you take positive numbers, you do NMF, and then you try to see what you get. That's best. But what is the real map between the signatures and the molecular biological processes? It doesn't, I mean, for me, it's not clear that this would be a one-to-one. I mean, they can be very different, and we see from the data in the experiments that it's a little bit more complex than that. What is more interesting is that we are discovering many signatures, so many axes, that we don't have any biological process that we know of that can explain that. Indeed, many of those, if you go to the database of Sanger, most of them, we don't have any biological process that can explain this particular flavor of mutations. But it's interesting. It's opening the question of trying to see if there is any other thing that we don't know. It can be endogenous or exogenous processes that can do to these things. And there is also something that is a bit more technical, so how we incorporate, for example, rearrangements or the hairpins that I was mentioning. So I will stop here, and then I can just continue briefly with the single cell data later.
Okay, should we come back at 2:15 or something?
Yes. Yes. Yeah, I'm going to stop and then.